This is Duke University.
Good afternoon. I'm Mary Klotman, Dean of the School of Medicine. Although I'm unable to join you today, I'm delighted to have the opportunity to introduce your distinguished keynote speaker. Once again, I want to congratulate the Duke Institute for Brain Sciences and Jerry Dawson and her team for an outstanding event. Each year, this lecture highlights outstanding science and scientists, and this year is no different. Earlier today, you heard from three of Duke's very own scientific stars, Nicole Kalakis, Palin Balkin, and Michael Tadros. And now it's most fitting, during Women's History Month, that we welcome today's speaker, Dr. Eve Marder. Dr. Marder is considered one of the most influential neuroscientists of her generation. She is the Victor and Gwendolyn Beinfeld Professor of Biology at Brandeis University, and for more than 25 years, her lab has combined experimental work with insights from modeling and theoretical studies. Among their many discoveries, her lab pioneered studies of homeostatic regulation of intrinsic membrane properties and stimulated work on the mechanisms by which brains remain stable while allowing for change during development and learning. Dr. Marder also helped develop a breakthrough in electrophysiologic method known as dynamic clamp, now used widely in laboratories around the world. Currently, her lab is studying the extent to which similar network performances can arise from different sets of underlying network parameters, opening up rigorous studies of the variations in the individual brains of normal, healthy animals. Known for her mentorship of students and early career researchers, Dr. Marder established one of the first undergraduate neuroscience programs in the US at Brandeis. She is a member of the National Academy of Science, the National Academy of Medicine, the American Academy of Arts and Sciences, and a fellow of the American Association for the Advancement of Science. Among her numerous honors, in 2016, she received the Kavli Prize in Neuroscience, and in 2019, the National Academy of Science awarded her the award in neuroscience. To say we are thrilled to have her at Duke is an understatement. Please join me in welcoming Dr. Marder.
It's really a great pleasure to be here or be there. I feel like I'm with you. And I would just like to make one comment, which is it's extremely wonderful to be in the company of so many really talented women scientists, because it shows how much Duke has really come into its own as an educational institution, in addition to all of its sports teams, etc. What I'd like to do today is hit on some of the themes that I think came up during the day, and I was really quite touched to pop into a poster session and see one of our slides, and then, and of course I missed the first part of that poster, so I apologize for that. But as I think you all know, what we've been doing in recent years is talking about differential resilience to perturbations of circuits with similar performance, and this really goes to many of the problems we've been talking about in the last 10 or 15 minutes, and a lot of what you were talking about all day. So before starting, I would just like to acknowledge the wonderful people with whom I've worked. My present lab is up here. It includes a number of postdocs and graduate students and a couple of undergraduates and technicians. It's not 25 years, it's 40-something years that I've had my lab. I've had the extraordinary privilege of working with a tremendous number of very, very talented people. And I would just like to make the point that I think a really good PI is someone who actually recognizes the genius and the creativity and the people in the lab. And everything really important or good we've done is because someone in the lab did something, thought something, and then brought it to me, and I was smart enough to realize they had done something really wonderful that I never would have thought of myself. So I really owe a tremendous debt of gratitude to all these people. Now, my lab alumni include people who are still postdocs who are doing all sorts of things, and then a large number of people have gone on either to industry, government, or faculty level positions. And I'm equally proud of the people who are now program officers at the NIH and the NSF, or who are faculty or who work in industry, because that wonderful training that a PhD gives you is really important for all aspects of our modern scientific life. And I'm really quite privileged to have worked with so many wonderful people. Now, the problem that I'm going to be talking with you today about is the one we've really been talking about all afternoon, which is how is individual variability related to resilience to challenge? And obviously, and I made this slide before this conversation, this is particularly relevant to the effects of stressors on mental illness and on the variable efficacies of therapeutic agents on mental illness. And I think we're so far from really understanding how to select therapeutics in terms of the underlying individual variability, but hopefully the work that many of you guys are doing will bring us there in the future. Although I'm not sure I'm going to live long enough to see this that successfully, but hopefully you'll do it anyway. Now, most of you know that my entire scientific career, I've worked on the small nervous system, the crustacean stomatogastric nervous system, and what I have done is by working on a small nervous system, one with individually identifiable neurons, it's allowed us to see and pose questions that are certainly general to all nervous systems, but are just easier to articulate when you can go back to the same neuron from animal to animal. Now, all the data I'm probably going to show you today comes from the pyloric rhythm of lobsters and crabs, mostly crabs recently. And I'd just like to tell you what we do to make these recordings. So the animals we work on live out in the North Atlantic Ocean, right outside of Boston, where I live. The animals are wild caught, so they're a much better model system for the human population than are your genetically inbred model organisms in the lab, because they have all of the idiosyncrasies of the human population as they have been out living in the ocean. The crabs and lobsters we work on are 5 to 7 years old, which means that they've dealt with the vagaries and the hostility of the natural environment for that period of time, and that I think will become important later. The fishermen catch the animals. They will never tell us where they've caught them. And therefore, we actually don't know the individual natural history of the animals, although I would really love to know that. We once tried to bribe a fisherman to measure the temperature and the depth which he was catching his animals, and he did it once, and then he took the sensors we had given him and he disappeared. So I think that's not that unusual. So the animals go first to a distributor on the waterfront. And then in the old days, they used to take a taxi and come out to campus. Since Brandeis will not allow taxi drivers on campus any longer now, one of my technicians goes down to the harbor and brings the animals back. Then they go down to seawater tanks in the basement of our lab and basement of our building. And then they come upstairs and we dissect the stomatogastric nervous system from the animal. Now, the stomatogastric nervous system controls the gut or the stomach of the crab or the lobster, but the stomach of a crab or a lobster is completely different from a human stomach. This is a system that's much more like your mouth parts or your shoulder, as there are 40 sets of striated muscles in the stomach, and they are moved by a series of motor neurons that move these striated muscles. So as I said, it's really a locomotory-like system, and in some ways it's very much like your respiratory system. So what we do is we take the stomach, I used to, they do, take the stomach out of the animal, dissect the nervous system into a Solgard-lined petri dish, and then we superfuse it with physiological saline, and then we record either intracellularly using sharp microelectrodes from 3, 4, or 5 neurons individually or simultaneously, and then we record extracellular from the motor nerves. Now, the motor nerves would have carried the axons that would have gone to the muscles if the muscles were still there. And it's important for you to know, as I show you the activities of individual neurons, those activities these neurons are identified absolutely unambiguously on the basis of what their projection patterns were. And I think it's very, very important in all of the work we've done over all these years. I really have come to deeply believe that the best identification, if you have a single criterion, it's the projection pattern, and then physiological signatures are second best, and molecular signatures are third best. And obviously the best tool is combining all three. Okay, so what do we see? Here you can see the pyloric rhythm, as we call it. And what it is, it's a triphasic motor pattern. You can see it here as LP, PY, PD, activity in LP, PY, PD. And this is in the simultaneous intracellular recordings. And then extracellular recordings, you're seeing those same elements on a nerve that actually carries the axon of all three of those neurons. And so you see LP, PY, PD, LP, PY, PD, and the time bar is missing here, but this is about a 1 Hertz rhythm at 11 degrees, which is our control temperature. Now, to the right, you see what we used to call a wiring diagram or a connectivity diagram. Today, we would call that a connectome. And that is to say, it tells you how everything is connected. Now, when I was starting in the field, the holy grail was to come up with these wiring diagrams for small circuits. And the hope was, my thesis advisor believed, that once you had a wiring diagram, you'd understand how it worked. And that was really the goal in the 60s and the 70s. And by 1980, we had this connectivity diagram or wiring diagram, and it became patently clear that the connectome itself was absolutely necessary and completely insufficient to explain the dynamics. In fact, if I showed you this wiring diagram and didn't show you the dynamics, there's absolutely no way you would know what this thing was doing. Now, I can tell you, and by 1980, we had a first generation explanation of how this works. And the simple answer is that there are these 3 neurons. The AB is a bursting pacemaker. It's electrically coupled to the 2 BD neurons, and therefore the 3 of them fire together in bursts. These are a pacemaker kernel for the pyloric rhythm. The AB and the PD neurons together rhythmically inhibit the LP and the PY neurons. So the LP and the PY neurons fire on rebound from inhibition. All of these synaptic connections are inhibitory. And then there are also, you can see, half-center oscillators or reciprocal inhibitory connections between LP and PD and LP and PY. So with the way the rhythm is generated is you have a pacemaker kernel, LP and PY are shut off. LP recovers from its inhibition first. It keeps PY down. PY eventually recovers from inhibition and turns off LP, and then the whole thing starts again. Now, what we set out to do after we had this connectivity diagram was really trying to explain in terms of detailed cellular biophysical mechanisms how this connectivity diagram gave rise to these dynamics and then to the modulated dynamics that we were starting to study. Now, it becomes patently clear that all the information that's missing in this wiring diagram is you need to know, if you really want to be able to go from the connectivity to the dynamics, you need to know the nature of the voltage and time-dependent currents in each one of these cells. You need to know the strength and time course of all the synapses, and then you need a way of putting all that information back together again. And that's why by the end of the 1980s, I was starting to collaborate with theorists, because it was very clear that the reductionist pathway I was working on, if you really wanted to be able to account for system behavior in terms of individual biophysical mechanisms, you have to have some way of putting all the story back together again. And that becomes sort of theory. Now, what I'm going to do today is show you a lot of models and a lot of insights that can come from looking at simple models. And these models generally have the same form. So I'm going to show you what it is. We use single compartment models with usually 8 conductances and a capacitance. So these models are far more complex than many of the models that people who work in large circuits use, but they're still a vast oversimplification when compared to what a real neuron looks like. So in these models have a single neuron with obviously the lipid, that's the capacitance, an inward sodium current, 2 inward calcium currents, a low and high threshold, sustained and transient ones, an IH, that's a hyperpolarization activated inward current, and 3 potassium currents, an 8 current, that's the transient outward current, calcium activated potassium current, and the classical delayed rectifier current. So as I said, this is a model of intermediate complexity. It's biologically realistic in that we implement it in a conductance-based fashion. That is a lot the way in which real neurons work, but still it's missing all the details of the structure that we all know and love. So the first thing I want to show you is a technique, a way of visualizing things that one of my postdocs recently developed that is tremendously helpful to us. And so what this is Leandro Alonso, what he wanted to do was find a way of displaying, now this is a model's behavior, this is voltage as a function of time, of a single neuron that's meant to capture the dynamics of that pacemaker kernel. So you can see it's hyperpolarized and it depolarizes, fires a burst of action potentials, etc. And one of the things we always want to know is what's happening to all of the currents at any moment in time. And so that's what I'm going to show you. What Leandro's doing here is he's showing us the outward currents plotted up as a biophysicist would, and inward currents plotted down. This black bar here is the total outward current on a very compressed log scale. And the total inward current, again, on that compressed log scale. And now in colors, what you're seeing is the percent of the total outward current in purple, shown with the A current, the calcium activated potassium current in orange, the delayed rectifier here in yellow, and the leak, I think, is in pink. Down here in the inward currents, you're seeing the sodium current in red, and the 2 calcium currents in green and blue, and the H current in brown. So this allows us to really visualize how the cell is producing its behavior. And this then becomes a tool that I'm going to use to show you to make the first point, which I think many of you know, because we've been talking about this for quite a long time, that there are multiple solutions to producing very similar behavior. Now this slide looks a lot busier than it actually is, so I'm going to talk you through it. What Leandro did here is he's showing you 6 model neurons, 6 bursters with different sets of parameters. They have the same 8 currents, but different amounts of each, or different maximal conductances. So here's neuron A, B, C, D, E, F. You can see that the dynamics of the cell to your eye look very, very similar. On the other hand, if you now look at the currents, if you look at the A current in purple, it's very small in model A, and it's humongous in model F, and it's pretty big in model E, and you can see all different amounts. Here in orange is the calcium activated potassium, and you can see that it's, again, changing amplitude or changing the amount of participation in the total outward current that you see. So although the dynamics look very similar, these models are degenerate in that they have very different sets of, in a sense, mechanisms that are giving rise to this burst. And you can see all the other currents are changing as well. The importance of the H current is changing, green, you see is changing, etc. And here are the actual maximal conductances. Now, I'd just like to make one more point about this before we move on, and which is in purple, the A current, which is humongous here. It turns out that the maximal conductance of the A current, which is up here somewhere, it's being hidden by, the maximal conductance is bigger in E than it is in F, even though the current is bigger in F, and that's because of the expression of all the other currents. Okay, so this is our first instance of seeing multiple solutions. Now, many of you know we've been working on this concept for quite a while, and many of you know the paper that I saw on a poster today from Astrid Prince, which really started us on this whole line of endeavor. So this is a paper that was now published quite a while ago. And what Astrid, who was then a postdoc and is now at Emory as a faculty member, what Astrid wanted to do was to build a successful model of the pyloric rhythm. And she started out trying to just find what she considered good models for the PD cell or the LP cell or the PY cell. And she started out with some models we had floating around the lab, and she hand tuned for about a week, and she found it extremely disconcerting because it is actually extremely hard to hand tune a conductance space model, because the minute you change one thing, something else changes. And it's so instead of just fiddling, Astrid said no, and she just built a database of 1.7 million model neurons, where she chose 6 values for each of the 8 conductances in the model. And then she ran them all, and then she selected them to find what she considered good models for the individual cell types. So she found good models for the ABPD, and good models for the LP, and good models for the PY. And then she took those, and she then simulated 20 million versions of the pyloric rhythm with all different kinds of parameters, and then selected from those ones that gave good models of the pyloric rhythm. So from the 20 million starting, she ended up with 400,000 that she considered good pyloric network models. And obviously they were all different because they had different sets of parameters. When we were writing the paper, I just told her to find 2 model networks with pretty similar behavior and different conductances. And she came back with this particular picture 2 hours later, so you can see how easy it was for her to do that. So model network number 1, model network 2 have very similar behavior. On the other hand, if we look inside them at the underlying conductances, and here are just some of them. This is the H current in the PY. It's big here, it's small here. The PY to LP synapse is big in this model and small in this one. The AB to LP synapse is big in this model and small in this one. So you can see this set of parameters and this set of parameters are totally different, and yet they're together producing very, very similar behavior. So I was incredibly excited when I saw this. This is one of the things I said that, you know, wonderful people in your lab may do things you never would have thought to do. This was one of them. And this work of Astrid's really set us off into a whole lot of new directions. But one of the reasons I was so incredibly excited was because we had done all kinds of measurements and seen a lot of variability in the biological measurements we were making from animal to animal. And one always worries when you see variance in your data, whether it's your fault or whether it's true. And this immediately said that you might expect to see a lot of variance in the measurements you're making, because if lots of solutions are possible, then why shouldn't the crabs have found them? So when I started talking about this, I took a lot of flack from a bunch of theorists. And the flack came in two major kinds. There were some theorists who told me that this had to be wrong because everybody knew that biological systems were all optimal. And I would look at them and say, since when are you optimal? And that would tend to shut down that particular sort of line of direction. But what they did say that was really important, and it really, really helped me frame it. They said, yes, this set of parameters and this set of parameters are different, and yet you're seeing very similar outputs. But as soon as you perturb the network, you're going to see that this network and this network have to behave differently. And I started thinking about this, and it's certainly true that in the universe of all possible perturbations, this set of parameters and this set of parameters, there will be a perturbation that will separate them. And then I started thinking and saying to myself, well, yeah, there will be perturbations that will separate them, but the question is, are the perturbations that the animals will see in their natural life, will they be perturbations that will separate them? Or can imagine a crab with this set of parameters and a crab with this set of parameters, can they be nonetheless robust and give reliable responses to the perturbations that they're going to see in their life. And if not, how will they break down or how will they crash? So that is what set us off on what became the next 15 years of the work in my lab. And we set out to answer these questions: how variable the parameters in real biological networks? Do perturbations reveal cryptic animal to animal differences? How reliably can animals with intrinsic variability respond to perturbations? And this is a question we basically know the answers to these questions. And then, are some sets of parameters associated with higher resilience? And this is something that I think, in a sense, we were hinting at today during the other talks. We would like to know exactly which set of interacting physiological parameters are associated with higher resilience or higher resilience in the face of certain kinds of stressors. So very quickly to go through, we set out to look at the variability of a number of things in the real network. So these are now biological data. Each animal has only one LP neuron. So this is LP neuron from animal 1. This is LP neuron from animal 2. They're both in the network. They're firing very similarly, and you can see how similarly. And this is work that Jean-Marc Gaillard and Dave Schultz did. And then Jean-Marc took these LPs and he went into 2-electrode voltage clamp, and then he measured those 3 potassium currents that we're talking about. Here's the calcium activated K in LP2. Here's the calcium activated K in animal 1. And you can see it's much bigger in animal 2. Here's the A current in animal 2. Here's the LP in animal 1. And delayed rectifiers are slightly bigger here, but much less so than the other currents. And as we measure them, as Jean-Marc measured them, now we're seeing the conductances at measured, actually it's the conductance densities. In voltage clamp, you basically see a 2 to 5-fold or 2 to 6-fold range for each of the currents. And these data are very similar to data that Jorge Golowash had obtained years prior. And it's always nice when a new postdoc recovers the same data as a previous postdoc. But the new thing that Dave Schultz added, after Jean-Marc had done the voltage clamp, Dave Schultz hand dissected the neurons, and now we're looking at single neuron from which the physiology had been, the biophysics had been done. And now we're looking at the mRNA copy number for SHAB, BKKCA, and SHAL. That's one of the genes for the delayed rectifier, calcium activated K, and one of the A current genes. And you can see, again, there's this 2 to 5, 2 to 6-fold variability. But interestingly, because we had the data on the voltage clamp and the molecular data together, when Dave plotted the conductance and the copy number, you can see there's a very strong correlation between the conductances we measured in voltage clamp and the copy number from the molecular work. And I think obviously this doesn't have to be, the protein and the mRNA don't have to be that tightly correlated, but for many of our channel genes in these cells, they are. And I think that's partially because these animals live in the cold water and channels turn over relatively slowly. So I'm not sure this would necessarily be true if we were working on warm-blooded animals, but it is true. Now, more recently, Dave Schultz has done a much more extensive analysis of channel densities in those same identified neurons and every identified neuron in the semenogastric ganglion. And now we're seeing, now I think we have 20 or 22 different channel genes. And again, you see this 2 to 5, 2 to 6-fold variability for all these. Now, some of these are the ones we previously studied. All the big ones are ones that we had seen in our first studies. And added to that, there's some that are much lower, lower amplitude signals. But you see that the same neuron, the same individual neurons, have very large numbers of channel genes. And we're going to come back to this later. Okay, the other thing that we discovered, or that Dave Schultz and Jean-Marc discovered is that there's some very strong correlations in the expression of the molecular data. So for example, IH, that's the HCN gene, is strongly correlated with shell, that's IA, in virtually every cell type, and where there are different sets of correlations in different cells. And we find that there's actually a set of cell specific or cell type specific sets of correlations in ion channel expression. Even though they all express the same channel genes, the correlated expression is cell type specific. And we can come back and talk about that later. Now, at the same time, we're seeing this 2 to 6-fold variability in expression. If you now record, and this is a slide that Adrian Topolik made just recently, just to make this point, these are extracellular recordings from 5 animals showing the pyloric rhythm, and you can see how similar they are. So despite the fact that there's this fairly substantial variability at the parameter level, at the overall network level, there's much more reliability or stereotypy. And that's seen here, the extracellular recordings, and the same thing is seen in intracellular recordings. These are the PD cells from 3 animals, and you can see how similar their waveforms look, the LP and the PY in 3 animals. And so despite that underlying variability, you can see the behavior of the cells in the network is very, very similar. So just to summarize where we are now, individual synaptic and intrinsic parameters can vary 2 to 6-fold across animals, even when they have very similar behavior. Now, what this immediately said to me is that we had to take seriously that observation or that prediction that the original grumpy theorists were making, that presumably these animals, if they have this kind of variability, that perturbations might affect the individuals quite differently. So we set out to look for physiologically relevant perturbations. And we've spent a lot of time studying temperature, pH, and then high potassium. Temperature and pH are two perturbations that the animals have to deal with all the time. And high potassium is something that many of our cells deal with all the time, and that physiologists use as a perturbation. Now I'm going to spend a fair bit of time talking about temperature, but very similar data are seen with pH and high potassium, except we have a new story with high potassium, which goes to the issue of adaptation, which we see actually a very interesting set of adaptations to sustained or repeated applications of high potassium. But I won't have time to go through that right now. Okay, we'll go to temperature. Now, temperature is obviously influences every animal, all of us, but most importantly, an animal that lives in the ocean, in the North Atlantic Ocean. The ocean temperature where our crabs live can vary from about 2 or 3 degrees up to about 25 degrees from winter to summer. And in a given day, a crab might see a 10-degree fluctuation of temperature as tides come in and out, as currents come in and out, as weather happens, etc. So when I was in college, I was taught that a Q10 was a way of telling you how temperature sensitive a process was, a Q10 of 2. And I was told that biological systems all had Q10s of 2. Well, of course they don't. But a Q10 of 2 is a doubling of a process in a 10-degree centigrade. A Q10 of 1 is complete temperature invariant. A Q10 of 3, you can see the exponential. Now, I should just tell you ahead of time that most ion channels, their conductance and their activation and inactivation rates of opening and closing of channels are in the range of about 1.5 to about 4. But there are channels that are used for temperature sensing that have Q10s that can be as high as 20 or 40 or 100. But in general, most Q10s are in the 1.5 to 4 range. Now, if you think about it, if you do a really fast thought experiment in your head, you'd say, well, okay, if I've got a neuron with 8 or 10 different voltage time dependent channels, and it's got to generate a bursting set of mechanisms, if all those channels have the same identical Q10 for every single one of the processes, should be no problem. But if the Q10s are all different, you might imagine that the dynamics would get out of whack as you start changing temperature. And that's what you can see here. This is a simulation that Tim O'Leary, who was then a postdoc, made just to make this point for a paper we're working on. So here are 3 models which have very, very similar behavior at 10 degrees, but each of the models has a different set of Q10s. And so this model here, when you warm it up, it goes from being a burster to a faster burster, and eventually looks like a single spike burster. When you cool it, it goes to a slower and slower burster. However, this model, which looks very similar, becomes a single spike burster and then goes into depolarization block at high temperatures, and it becomes irregular at low temperatures. This model, interestingly, becomes a single spike burster when you cool it, and then crashes in a hyperpolarized fashion when you warm it up. So these 3 cells show you none of them is particularly robust to temperature, but they are crashing in a very different way. So now what about my animals? And Lamont Tang, who was a graduate student at the time, did these experiments. Here we see extracellular recordings from crab at 7 degrees. So we have PD, LP, PY, PD, LP, PY at 7, and now at 19. You can see a beautiful triphasic rhythm, and it's faster as you would expect. And indeed, if you now plot the frequency of the network as a function of temperature, it has a Q10 and a parent Q10 of about 2.3. But what was remarkable about this study, which really blew my mind when Lamont left this on my desk, is that the phase relationships, that is to say, the normalized time of opening and closing of the ion channel genes, or the normalized time at which each cell would start and stop firing, was completely temperature conserved. So here's PD off. We count the start of the cycle of the first spike of the PD. So PD off, LP on, LP off. You can say that even though the frequency is increasing, the normalized time or the phase relationships are maintained. And if you think about it in a motor system, what matters is the time at which each muscle is activated relative to each other, even when the frequency is changing or the period is changing. So now remember, all these crabs have these different underlying structures. Nonetheless, now this is the same, basically the same message you saw in the previous slide, just done with the intracellular recordings, 7, 11, 15, 19, 23. Beautiful pyloric rhythm, increasing in frequency. And when Lamont superimposed the 7, 11, and 19 degree waveforms, they're perfectly superimposable, which says that even though all the processes are temperature sensitive and they all have different Q10s, nonetheless, they work together to maintain constant function. Okay, so by now you're thinking, well, what happens if we go to more extreme temperatures? Now, 23 degrees in 2009, when Lamont probably did this, 23 or 24 was what we used to think about as being sort of the high end of what the ocean would be in these animals' habitat. But Lamont then went to temperatures that were much higher, and he got what he called crashes. And so these are 4 different animals, A, B, C, D, 4 different animals. And you can see that at higher temperatures, I think these are about 27 degrees, each one of them is producing irregular behaviors, but they're all different. And the if you think about it, the prediction is that if each animal has a different set of solutions or a different set of parameters, you would expect them to crash differentially as you start pushing them. And so that's exactly what we were seeing. Now, however, and this gives you pause for the world we live in 2012, after we had published the first couple of papers about this, we had a winter that was extremely warm. And in fact, it was the water was 6 or 7 degrees warmer than normal throughout the whole winter. And the spring after that, Sarah Haddad, who was a new graduate student in the lab at the time, was able to take the animals up to 36 degrees and produce pyloric rhythms that were faster than we had ever seen before. And this actually was very disconcerting because we were seeing in our laboratory animals the effects of long-term climate change. In following years, the crash points went back down, and so we were able to crash animals, by 25 to 30 degrees as we had with Lamont. But the point I'm showing you here is, first of all, that 6 months of the ocean being too hot is definitely producing changes in the animal's behavior, which I hope concern you all. But the other thing that is really, really interesting is if I had just shown you the 11-degree traces, that's our normal physiological condition, we never would have suspected that the changes that took place had taken place. And so hidden in the control, unstressed condition were a lot of changes that were completely cryptic or non-transparent until we had done the stressor. Okay, now more recently, Leandro Alonso wanted to build some models of the effects of temperature because he wanted to start asking questions about the role of individual differences and the role of different currents in dynamics. And so what he set out to do is to find models that were well temperature compensated up to 25 degrees, and then would crash at 30 degrees. Now, it turns out that it's actually very difficult to find models that work well throughout this whole range, and we'll come talk about that. But what Leandro used is a very strong genetic algorithm, and he was able to find them. So here's a model at 10, 15, 20, 25 degrees. The frequency is increasing, the phase relationships remain constant, but then at 30 degrees, it's completely crashed. And he found a number of these models. So I'm going to show you now 5 of those models. This is on a compressed time basis. It's the same on a more spread out time base. 1, 2, 3, 4, 5 different ones. And if you look at the spread out time basis, you can see that they're all disordered, but they're all disordered to a different degree and in different ways. And that's because they, like the biological systems, all had different sets of conductances and parameters in the model. So again, you're seeing here the differential crashes of animals with different underlying, in this case, models of different underlying parameters. So the conclusions from these crashes were, at low temperatures, all networks are well behaved, showing consistent changes in frequency and outstanding temperature compensation. Each individual animal crashes with individual dynamics as predicted by their underlying parameter differences. I didn't have time to show you this, but Sarah Haddad did some very beautiful work that showed that some neuromodulators actually expand the stable range of temperatures, while others make the preparations more temperature sensitive. And this comes back to some of the issues we were talking before about what you can expect as a consequence of neuromodulator action. Now, I just have to show you a few more slides from these models before moving on. This is my favorite slide of 2020. So what Leandro's done here is he's showing you the last spike of a burst, and he's showing you with his current scape so you can see what's happening. So this is the same model at 10, 11, 12, 13, up to 25 degrees, and you're just seeing the last spike of the burst. And now if we look in the current scape, you can see here the A current is big at 10, And then as we go higher and higher and higher in temperature, you're seeing the A current is getting smaller and smaller. In contrast, the calcium activated K starts out small. It gets bigger and bigger and bigger and bigger as you go up in temperature. And the delayed rectifier is also changing its component, as are the other currents as well. But the reason I love this is because it's showing you that the mechanism by which the burst is terminated is entirely different here as it is here. And so what you're seeing is that the currents, the 3 potassium currents in this model, are tiling in voltage and time so that the cell can smoothly make a transition, a stable transition across the perturbation, across the temperature range. So this mechanism and this mechanism, you have the cell making these smooth transitions and trading off. The role of these K currents in spike polarization is changing very, very smoothly. And I think this is one of the reasons why the reason I like this result so much is I think this answers one of the fundamental questions of why all of our neurons have so many different currents. And I think they have so many currents precisely so that you can actually be unlimited by going through crashes. You can sort of protect against crash. This gives you increased resilience because you're substituting the behavior of one channel for another. Okay, now just a couple more observations with these. Here are 2 models at 10 degrees and 25 degrees, model 1, model 2. And now what Leandro's doing is he's just showing you, here's the voltage. It's getting faster. Now he's plotting the same current at 10 and at 25, the same current is getting bigger in this model. In this model, the same current is getting smaller as you go from 10 to 25. Here you've got one of the calcium currents it's much larger as you go from 10 to 25. In this model, the calcium current gets smaller. And if you look, you can see here you've got the calcium activated K is tiny at 10, it gets really large, 25. And here it changes, gets slightly smaller as we go. So these 2 models both can operate at 10 degrees and at 25 degrees. But as they make those transitions, the role or the contribution of each of the currents in the dynamics changes in ways that are completely different. And I think as we think about people and about individual differences, it's important to keep this sort of kind of image in mind because it gives you a sense of how individual variation can really tell you that very different kinds of changes can be responsible for pretty robust behavior. And then pharmacology, while we're on pharmacology, here's a model, which now we have a network model with 100% of the A current present at 10 degrees. And then when Leandro turns off the A current, blocks it effectively. And now you can see the network is completely disrupted. However, when he does the same thing at 25 degrees, here's the 100%, and now the rhythm is not particularly sensitive to turning off the A current at 25 degrees. So therapeutically, a drug that blocked the A current would be very, very effective at 10 degrees and very ineffective at 25 degrees. Or it would be robust at 25, the same individual, and not at 10. And here's the opposite result. Here's the calcium activated K at 100% at 10 degrees. When you turn off the calcium activated K, the network slows down a little bit, but the rhythm is maintained. However, if you do the same manipulation at 25 degrees, you can see the rhythm is completely sensitive. So obviously, most human networks don't operate at 10 degrees and at 25 degrees. But on the other hand, this tells you that the starting state of a network can completely alter its pharmacological sensitivity. And you guys all know that, but this allows you to see it in very, very, very simple and discrete terms, that depending on your starting set of conditions, that is in this case, which is expressed by temperature, you can completely alter the effectiveness of a pharmacological agent. So individuals with similar starting circuit performance with relatively similar circuit mechanisms in the control state can nonetheless show entirely different crash mechanisms. And I think this, obviously you all know, this is a cautionary tale for thinking about disease. Now, I think I have a couple more minutes, so I'd like to very quickly talk to you a tiny bit about finding these models. Because if you randomly sample parameter space, you can't, it's really difficult to find neurons that are temperature compensated. They're not common in the population. We had to search large numbers of models to find them. On the other hand, every single crab has found a solution by which it can live in the ocean. And so the question is, what rules could biological systems have that allow them to find neurons that are well temperature compensated, to find some of those subset, crabs can have all different kinds of solutions with multiple different sets of parameters, but not all parameters work. And even in Astrid's original work, only 2% of those solutions that she studied were successful. And so the question is, if random association of parameters are so unsuccessful, how do neurons and networks find the successful ones? So we have been studying homeostasis or mechanisms of homeostasis for many, many years. We started out thinking about it because neurons will live for 100 years, but receptors and ion channels turn over in hours or days or weeks. And so you have to think about function being maintained while the nervous system is constantly rebuilding itself. And so in these homeostatic models, we stipulate that neurons can self-tune their conductance densities to maintain a target activity. And this is quite different from saying they're going to control the number of any one kind of channel, but they're going to control all different kinds of channels in different ways to maintain a target activity. Now, in all of these models, and we've built many generations of them, the idea is that you have some sort of sensor of a target activity. In the first set of models, it was a intracellular calcium. And then that we've gone through several different classes of these models. But this just reminds me to tell you, I don't know if this is in the wrong place, but brains are not stationary. And I think it's really important for us all to remember that brains are constantly rebuilding themselves, replacing every component continuously. And then I guess this slide I made for a different talk, but this raises the conundrum. Has the brain function, memory, and sense of self persists when all brain components are in a constant state of flux? But just want to show you a little bit of the answer to the temperature compensation. And this comes from a new class of homeostatic models that Tim O'Leary built when he was a postdoc. And in this model, and this is the third generation of these homeostatic models, Tim is saying that there'll be a turnover of each channel type in a calcium-dependent fashion, but the turnover rate or the expression time constant is different for every type of ion channel. And so, and there's a target calcium concentration, an actual calcium concentration, and then the channel density has a turnover that depends on that. So if we can just look at sort of self-assembly of these models, if this is random initial conditions, an initial starting place, when you run the model, if you set it to have a target of a burster, it'll become a burster. And here we have the same sets of conductances, and we grow ourselves to a target activity pattern. Importantly, if you do multiple runs for different initial conditions, you actually get multiple models with very similar behavior, but with different underlying conductances, because this is a very compressed log scale. And then you're recovering this 2 to 6 or 2 to 10-fold range in each of the parameters living inside of these 6 models. But as you change the regulation rates for each of the currents, you get different kinds of cell types, and you end up developing a whole bunch of of cell type specific correlations in channel expression. And these are the correlations that Tim was seeing in his model. And these correlations look very similar to the correlations we saw in our biological data at the mRNA level. And then using that, Tim was able to work backwards from these correlations in this kind of model and easily find models that were temperature robust. So here you've got 1, 2, 3, 4, 5 different of those temperature robust models that basically were easy to find by knowing which correlations were found in good bursting neurons. So the way we think about this now is that the crab must have built in a set of correlated expression of certain kinds of ion channel genes. That doesn't mean to say that so that IH and IA will always be correlated within an animal. Across animals, they can vary in that 2 to 6 or 2 to 8-fold range, but IH and IA are held together, as are some of the others. And then that allows each of the animals out there in the world to assemble itself into neurons and networks that work. So I'd just like to summarize this at the end. Correlations arise automatically in self-tuning homeostatic models and depend on the rates of the tuning process. These models generate variability in multiple cell types. And I didn't show you this, but these homeostatic processes may be working correctly and yet produce pathology in response to some perturbations. And we can use these models to find neurons with desired sets of properties like temperature, variance of duty cycle, etc. Now, we still have some additional new sets of homeostatic models. So we're continuing to explore ways in which to understand differential resilience and robustness. And like just to summarize at the end, differential resilience in the population is a necessary feature that comes from the inherent degeneracy of multiple solutions to producing similar behavior in healthy nervous systems. Some mechanisms for differential resilience are automatic and arise from the properties of ion channels, receptors, and signaling. And some mechanisms for resilience will depend on homeostatic mechanisms, behavioral adaptation, and learning. So I think it's important for you to realize that the temperature resilience that we see is basically built in, but then other mechanisms will require much longer term changes. And I would just like to stop there and thank you for your attention.
One of my favorite speakers. I always take away something new and think of something different besides the incredible visual. Thank you, Eve.
Oh, thank you.
That was just remarkable. Just the entire program of research and how you've mapped it out over the years is stunning, truly.
Well, thank you.
So we have a question from Anita Disney. When circuits are doing homeostatic control of variables other than conductance, such as levels of non-synaptic modulators, what do you think is the controlled variable sensor and the means by which the control loop is closed?
I think, so one of my postdocs now is worrying about this a lot. He's trying to say, what exactly is the homeostatic target? In a rhythmically, so the problem is, and I don't know the answer fully to that. In our system, in a rhythmically active system, it's pretty easy to say, or in your respiratory center, your heart, it's pretty easy to say, you know what the output should be. So building a homeostatic model to do that is pretty easy, right? I mean, we did it 20 years ago, and we've done it many times since. It's not that easy, but it's pretty easy. It's much more challenging to say, how do you build a homeostatic model to tune a sensory neuron to show the right firing rate in response to a stretch, or the right response in terms of a change in temperature, right? So, and to do that, you have to ask, how does a neuron measure its own function? Where's the error signal? And how does it tune to an error signal if it doesn't experience the perturbation enough, right? So we don't fully understand that. This goes back to the question about the homeostasis and neuromodulator. I think the homeostasis with neuromodulator is a little bit easier in a way, because in our system, we have 50 or 100 different modulators if you look at them, but many of them converge onto the same currents, and many of them have these sort of interesting set of convergent and synergistic effects. So it's many fewer free parameters than you would have thought about, and many of them have saturating actions. They're built in ways in which the modulators don't push you to crash. In other words, it's very few of the modulators that we work on actually push you to crash the way the high temperature pushes you to crash, or the way pH 5 pushes you to crash. I didn't talk about pH, but this network functions perfectly well between pH 6 and pH 10, which is dramatically ridiculous if you think about it. But eventually, if you go to pH 5 or if you go to pH 11, they do crash. But most modulators don't crash the network, even at very high concentrations, because the modulators actually push you around to maintain more stable function more often than not. So getting the modulator concentrations right, I don't think is much of a challenge as trying to figure out how to build a sensory neuron that'll respond to stretch correctly. But then maybe what happens is, maybe that's a long loop control. The sensory neuron responds to stretch and it makes too many action potentials, something else comes back, and it could be a long loop control loop where everything is tuned via multiple loops.
Right, absolutely. So we've got a related question from Henry. You explored perturbations of your pyloric rhythm models with respect to changes in parameters like temperature, but also of interest is how a dynamical model responds to a perturbation in time. If the state of the system undergoes a kick, what kind of transient occurs as the system supposedly recovers its original dynamics, but maybe with different phase? Have you explored this and how transients to the rhythm depend on the choice of the model?
So that's one of the things that Leandro's doing right now, is modeling the effects of Marder, is one of my graduate students, has done this very beautiful work on high potassium, and I just very briefly alluded to. So what high potassium does is you put it on the network first stops, and then in continued high potassium, it recovers its function, and it sit there. It's not completely normal, but it'll be functional. And then you wash the potassium out and it'll come back to normal. And then if you do that 3 or 4 times, the time it takes for it to recover decreases. So it has a long-term adaptation that obviously has a memory of its previous perturbations. And so what Leandro's doing now is he's building a homeostatic model that actually shows that long-term adaptation to these transient kicks. I mean, in a sense, it's that same question that your audience member asked. So in that case, there's the network itself shows a slow adaptation to the perturbation, and then we have homeostatic models that basically do the same thing. And what Leandro actually did to get these models to do it, it's very interesting. What he did is he turns the homeostatic process on and off. So when it's stable and close to target, he actually turns down the gain of the homeostatic mechanism. And then when there's an extreme perturbation, he turns up the gain on the mechanism so it adapts quickly. And so the notion that you actually turn the gain up and down on the homeostatic mechanism allows you to think about these on and off perturbations. So that's why they're writing a paper right now. And I think that's actually a very, very interesting idea, that if you're sitting if you're sitting and you're perfectly stable, your heart is perfectly stable and it responds okay and nothing's wrong, then you basically don't have to do very much. You can let everything go sort of in the background. But if something's really wrong, then you've got to kick in and try to fix it.
Yep. So we can modulate regulate the regulatory mechanism, in other words.
Yeah. So, and what he's done in the model is just basically gating it on and off, or turning its rate of being on.
All right. We have another question from Josh. The correlated variations of ion channels that nonetheless achieve the same spiking pattern in the same cell type from different individual crabs suggest a coherent transcription regulation. So have you ever thought about looking at the transcription factor expression in the same cell type from different individuals?
So I just heard a talk the other day and I asked that person that question. It would be, if I were working in flies or a mouse, it would be a lot easier to do that than if we're working in crabs. So we have transcriptomes and we have sort of a fairly decent genome, but I don't know that we have transcription factors. I mean, I have to make a lot of guesses. So, it's the wrong, yeah, I mean, in my mind, that's how it works, right? But I don't have the transcription factors all worked out. I can make guesses looking at flies and say, so we could do that. We could think about that. But right, so far we haven't done it. But that's the model in my mind, that there'll be correlated transcription factors.
Yeah, it makes sense.
Yeah.
Well, I'd love to circle back to the question that came up at the end of the panel discussion about sort of pharmacological tolerance and the waning effects of medications, and how this complementary groupings of different molecules working in concert might help us to either wean people off different medications or maintain efficacy when a person needs it to function in life.
So I guess the real question is, which therapeutic mechanisms are going to push homeostasis in the right direction or the wrong direction? So for example, I don't know if any of you have ever had experience with beta blockers, but many years ago, I had an aortic valve replaced, and they immediately threw me on really high doses of beta blockers. And I ended up developing really nasty arrhythmias, which I kept on telling my cardiologist, it's the beta blocker that's doing it, it's not me. And after 3 months, I won, and I weaned off the beta blockers and the arrhythmias went away. So that's a case in point, is they were giving me a drug to prevent all sorts of bad sprouting. And the drug itself created an arrhythmia because it had my heart rate too low, that then I had to get rid of the drug in order to go back to normal. So that was reversible. That was a case in point where it was completely reversible, but it didn't have to be. And I think it's very, very common that, for example, beta blockers generate arrhythmias. As they're given to prevent arrhythmias or to treat arrhythmias, and they generate arrhythmias as well in different individuals. So I don't know, that doesn't really answer your question, but I think it goes back to the issue of people have to be more, or physicians have to be more sensitive to which individuals are going to have paradoxical responses to any given pharmacological treatment, because it could be that in 95% of the cases, a beta blocker is a really good treatment for an arrhythmia, and 5% of the cases it kills them. Right? That's probably not a good odd, but I think it's actually fairly apt. It doesn't always kill them, but it does create problems.
And how do we acknowledge that that's a valuable lesson?
And the real problem is I don't know that you know how to predict which ahead of time.
Right,
so
right.
That's the real problem. What you would want is you would want to be able to measure something that would be predictive of whether you're going to be helped or hurt by drug X, right?
Mhm.
Whether Tegretol is going to help with that particular seizure disorder or hurt it or whatever. And right now, I think it's just trial and error, right? It's some amount, there are people, there are physicians with really good therapeutic intuition, so they must know something. And other than that, it's largely trial and error.
That's right. Yeah.
And then, of course, you know, my favorite thing is, you know, the drugs that are used for seizure, pain, schizophrenia, and depression. And you sort of say, okay, you're using the same drug for 4 or more completely different disorders. And that must tell you that both the drugs are dirty and none of them are doing what people think they're doing, because they're just pushing, if they're working, they're probably working by triggering some sort of homeostatic mechanism. And the fact that they're dirty may be important.
Yeah, that may be an advantage. Exactly.
It may be an advantage because, you know, I think that's another thing that I think is really important for people to think about. A lot of people have worked on the assumption that the best drug would be a very specific drug. But if you look at our models, I think you might argue that a very specific drug would be the worst thing. Because a very specific drug will be very idiosyncratic.
Yep.
Whereas a
Yeah, that
dirty
explains.
drug, a dirty drug, which works on 5 different kinds of ion channels, it doesn't maybe matter so much if it just pushes the network in the right direction. It doesn't, that individual variability gets sort of averaged out because all the multiple solutions that you're starting out from different multiple solutions with pretty similar behavior. So maybe the dirty drugs are more likely to be helpful precisely because they're dirty.
Could explain a lot of failures in drug development that we see over the years.
Well, I think so, because if you think about it, if you look at our models and you thought about it very seriously, you'd say the last thing you want is a highly specific drug. Unless you, right? Because I think, I mean, it's something we could actually ask very specifically, but I think it would be because, you know, if you look at the K channels, they're all working together, right? So maybe I guess if you have one that's really immune and that's giving you a real aberrant phenotype, maybe knock it out, but that's not the way cells work. So I don't know. I think we should just, that's why repurposing drugs that have been useful has probably been more effective than trying to get very, very selective new drugs. Because the repurposing, at least you know those repurposed drugs haven't killed that many people.
That's right. That's right. All right, here's a question from Jennifer. Is an inhibitory cell only model better suited for rhythm generation? And are there any of the features you've described today that are specific to inhibitory cell only model? In other words, is there any reason that these ideas can't be generalized to circuits comprised of other cell types as well? So is it limited to the inhibitory cells, or is it more generalizable?
I think all of these ideas are generalizable. In terms of small rhythmic circuits, pattern generating circuits, reciprocal inhibition is very commonly found in them, but most of them also have excitatory connections. I didn't talk about the modulatory control in the STG. The modulatory control is largely excitatory. The modulatory control in general upregulates the excitability of different cells in the network. There's nothing to say that this can't work in excitatory in networks with excitation. I mean, you know, everything that Gina Turgeon has ever done has been in cortical networks that work on excitation. So I think, you know, it'll play out, all these things will play out a little bit differently in each circuit. And I think that's another thing to remember, is that at some very global level, principles remain, but the details of how the principles get implemented in every part of the brain is going to be different and will depend on the actual cell types and properties of the cells there.
Yeah,
right,
that makes sense.
Yeah.
So for Eve, I really enjoyed the talk a lot.
Thank you.
I enjoy your style anecdotal thing, one of the first student projects I did when I was a graduate student was based on one of your papers. So, so do you, in terms of the different solutions that you see in crab, has anyone looked to see if that extends to the mammal? So for example, like dopamine neurons and cholinergic interneurons have a certain pace.
How would you know? The problem is, how would you know? I mean, I know it's the same. My intuition is it's the same. I mean, I think if you think about the respiratory center, which has to be very reliable and robust, I don't think we imagine that every human has the same number of prebiotic neurons in their respiratory center. I mean, somebody might have 79, another one have 89, or whatever the numbers are. So I'm assuming that this kind of animal-to-animal variability exists, but it's much hard. So data much like ours, you see in the leech, and you see in all the small systems people look, you see it. I'm assuming that it's true everywhere, but it's very hard to know what you would measure to see in a place where you have 10,000 neurons of the same cell type, where even if you do a connectome, you're talking about a probabilistic connectome, right? Not a one-to-one. I don't know if that answers your question.
Well, I mean, there's so many things to think about. So for example, in the crab, there's so few neurons in the STG that, the chance of there being a lot of animal-to-animal variability is high, because any given neuron is stochastically finding its solution, right? But then if you scale that up to 1 billion neurons that are each stochastically finding a solution, you might not have that much animal-to-animal variability anymore because of the numbers, right?
Well, you might have, or, I mean, it depends. If you think if you think those billion neurons are acting as an average, then maybe that's true. But if you think they're not acting as an average, but that they're working in slightly different collective behaviors, then it might be more. So I think it's very, it's a very model dependent statement you just made, because, if you think that 10,000 cortical cells with a variance of X can be captured as their average, I think I don't agree. So that's a conversation, because the minute you say you can capture the 10,000 as the average, and then you could collapse everything that the 10,000 are doing to one, it's not, first of all, I don't think it's true, but second of all, that's the premise of what you just said.
Yeah, that's fair. It depends on how
how you think the ensemble is working. Right. How you think the ensemble's working. So if the ensemble depends on the variable participation of individual elements, then it becomes much trickier to understand what that individual variability would look like.
Another question I had, and this is just a clarification, you showed covariances in the expression patterns, and it was pairwise. And I wasn't sure, is every pairwise, if you took any two channels and looked at them, do they always covary, or are there certain pairs that covary better than others?
No, there's some that covary only in one cell type. Okay. And there's some that don't covary at all. And there are others that have 3- and 4-dimensional covariations.
Okay. And are there certain kind of conceptual groupings? So for example, would 2 potassium channels covary typically, or would it often be a potassium against a sodium to balance each other?
We originally had thought, so all of the above, and the problem is, I mean, this is something we really have to do a much better job of looking at, but it's complicated. There's para and KD, there's this, that, and the other thing. IH and IA they're everywhere, but IH and IA is there all over the vertebrate brain too. So there's something very, very interesting about that very strong connection between IH and IA that you see in dopaminergic neurons, you see it in the hippocampus, you see it. So those two, it's pretty easy to see they control the approach to threshold after inhibition, and they work together to do that. Nicole.
Steve, I really loved thinking about the fluidity with which the potassium channels could step up for each other and beautiful functional redundancy. And just kind of pivoting to our conversation about when might you want a selective drug, I'm thinking that phenomena has been in such evolutionary pressure to have that functional redundancy. The human mutations that mess that up would create little vulnerability gaps. And so I was wondering from the modeling perspective, if you've ever had the opportunity to take a potassium channel that's known to have a pathogenic function, and whether it was its inactivation state or some other property, and show that now that creates this gap in the potassium channels taking over, like just in the somatogastric system, for example, you might be able to demonstrate that, even though it might have been in another system, a human system or something.
I know we could do that. I mean, we could take, I mean, there are all sorts of cases where you have people, like, inward rectifiers and stuff like that that mess up with long QT syndrome. I mean, there are lots of places where you have sets of channels that work perfectly well, fine until they don't.
And so in your, I mean, you have your three, yours that interdigitate, and it'd be very interesting if that ability, those mutations, when they were direct, the ones that were most analogous.
Probably the place that would be most instructive to do that would be in the heart, because we know a lot about the heart, and a lot about what mutations in the heart do. And they're right. And you've got, or maybe in some seizure disorder, some of the mutations that are seen in seizure disorders where, I find the things that I find most interesting are the things that people are walking around with perfectly fine for 20 years, and then they drop dead, right? Those are the ones, they've managed to stay away from those cusps, and then bam. So those are I can talk to my guys and see if they think about that.
The part I didn't share about the mechanism for the protein synthesis pathway is that we think it's changing the conductance of the SK channels, the calcium activated potassium.
Oh, cool.
And the cholinergic neuron is a little bit like some of your somatogastric bursters.
Yeah.
So I loved your picture of the orange, because I was like, that's what it's doing. That's why you just see it. The vulnerability right then. The basal rate isn't changed, but it's just this dopaminergic modulation, which is when that channel kicks in.
Cool.
For us in this neuron, it's what it uses.
Cool. That's very cool.
I'll just wrap things up here. I just, wow, what an afternoon, and just what a joy it is to bring together so many neuroscientists here at Duke. And I know that we had many people actually outside of Duke who are attending this, just coming together for an afternoon filled with just phenomenal science. And I know that for me, especially during this time that's been so challenging for all of us, it is science that has given us hope. And I think it is science that gives us hope really for understanding the complexities of the brain, and to watch the way all of you are dealing with this on such a mechanistic level now. And then how this is translating into a better understanding of how we can address, brain-related disorders. So just truly, truly inspiring. So I want to thank everybody for attending. I want to particularly thank Eve Marder for an absolutely inspiring talk.
My
Thank you
pleasure.
for coming, and perhaps you'll come and just be attendee next year, because you can be part of the discussion. But we will have another one of these events next year, hopefully in person,
For
if
you.
all goes well. So I'll just end by saying thank you again for
Thank
attending.
you all for well.
Yeah,
And we are all part of a giant project, experiment about what social isolation does to the brain. So there's easily 20 years of work to try and understand this perturbation in our lives.
indeed, indeed. All
Cross
right.
my fingers, 5 days will fix it. 5 days, right?
That's right. Yes,
days
we're
will
all
fix
going to hang on to that experiment.
5 days are gonna fix it in the normal, healthy individuals. And then there are gonna be some people who are,
Yes, and that's what we need to study, right? We need
Right.
to study that.
there are gonna be some
That's
people
resilience.
who've been,
That's resilience.
yep,
All
Okay.
right. Well, I wish you
Bye-bye,
all a
guys.
good evening. Thank you again.
bye-bye.
Take care, everyone.